again, very pleased to introduce Kyle. He's coming to us from the Department of Geography at UCLA, where he was recently hired. I think you said in November is when you showed up.
Mm-hmm.
And so Kyle and I have been running funny little circles around each other and never finally met until now. Kyle comes to us. He did his bachelor's at Trinity College in Texas, and then did his PhD at UC Santa Barbara, which is where I did my bachelor's degree work, with his advisor being Dave Siegel, who was my undergraduate advisor a long, long time ago, and I was actually in the geography department as well. And so that's just a way of just introducing you to what he's done. He has done a lot of work with remote sensing, both in mangrove systems and kelp forest systems. He has this really nice way of mixing remote sensing with an understanding of a species niche and statistical modeling to kind of give us some ideas about past, present, and future dynamics in this system. A lot of his most recent stuff has been done with giant kelp in the Santa Barbara Channel. I know this because he's published, even though he just got hired, he's published 8 papers, all in really good journals, 3 of which are in ecology that I was the editor for, really nice papers. And so it's quite clear that his skill set is being very well appreciated. I was looking through, he's done some time at the Smithsonian, he did time at Brown University, he did time at the CIA.
Yes.
Maybe
spying on kelp
looking
forests.
for terrorists in the kelp or something.
Yeah.
Pick up ISIS and GIS and kelp forests, I have no idea. There's also another program that Kyle's been working with that I've been involved with a bit, and that is he's a co-lead investigator with Jared Burns on the Zoonosis project, which is where they basically outsource to everyone on the planet who wants to help trace kelp canopies using the data they make available. So there's people out there on their own PCs or Macs or iPads, whatever they're doing, helping do the data analysis, which is, I think, a really awesome outreach program. And you guys can get on there yourselves and do it. And if you do a crappy job, they say, no, yours sucks. And they get it out of there. But it's really fun. And you see the kids when they get on there and start doing the traces. It's really a neat way to get people involved. And I think that comes, again, from Kyle's unique skill set and doing what he does. So we're really happy to have him here and listen to what he's going to talk to us about with giant kelp. Right afterwards, there's going to be a little bit more refreshments, and then people are going to head off to dinner. Kyle will be around tomorrow. Don't know the exact timing of when he'll be around, but he might be hanging out. So if you guys are at all interested in talking to him tomorrow, come to him after the seminar and set up a time, because he's staying right here at the Captain's Inn, and he'll be around till about midday tomorrow as well. So again, welcome, Kyle Cavanaugh.
Thank you. Thank you.
And thank you to Mike and Matt for inviting me and hosting me. It blows my mind that we have not met in person yet, and that this is my first visit to Moss Landing. As you'll probably see here, a lot of my research has been motivated and inspired by some of the work coming out of here, both in Mike's lab, some of his past grad students, and others. So I'm just super excited to be here. Thank you guys all. I'm looking forward to talking to you all individually. And as Mike said, I'll be around at least through midday tomorrow. So if you'd like to chat, if you're available tomorrow, please come up and see me. Unless you're a foreign national, then I have to report you to the CIA. Just kidding. So before I get rolling here, I'm joking about that. I'm not affiliated with them anymore. Before I get started here, I do need to acknowledge my co-authors and collaborators. The stuff I'm going to present today has very much been a group effort over the past few years. I want to, in particularly, highlight and acknowledge Tom Bell. He's a current PhD student at UC Santa Barbara. He was instrumental in building this huge satellite-based kelp observational data set that is at the center of a lot of what I'm going to talk about today. He's now taking this data set and done some interesting biogeographic analyses that I'll touch on too. And then Max Castorani is currently a postdoc also at UCSB, and he's built on some of our metapopulation work. I'm going to also highlight some of his recent research here. So I want to start my talk with a confession. So about 10 years ago, I started grad school at UCSB without a very good idea of what I wanted to do for my dissertation. And for any of you considering doing a PhD, I do not recommend this approach at all. But anyway, I started at UCSB with a pretty solid technical skills in remote sensing and GIS. I had some ideas, but I didn't know specifically what system I wanted to study for my dissertation. Then during the winter of that first year, Santa Barbara got hit by one of these large west-northwest swell events. All the surfers called it Big Wednesday. You know, these things always seem to happen on Wednesdays for some reason. But I was walking, you know, a day or two after that swell hit. I was walking on the beach right off the coast of UCSB, and this is sort of the scene that I came upon. And this blew my mind for someone not from California, not familiar with kelp forests. It was incredible. You know, the beach was littered with giant kelp that apparently had been ripped up from the seafloor and then deposited on the coastline. And what struck me was the scale of this disturbance, right? So to me, this seemed like the marine equivalent of some big terrestrial disturbance like a forest fire or a flood or something like that. But with those terrestrial disturbances, you often have a pretty accurate picture of the scale of that disturbance. So for example, with forest fires, it's common to know burned area. But I talked to a number of folks at UCSB, and no one really had a good quantitative idea of, you know, the scale of this, how much kelp was lost. And so this naturally piqued my curiosity, right? How many kilograms or tons of kelp had been removed here? How many square meters of kelp canopy had been lost? Was this an abnormal event for Santa Barbara? Over the course of a normal winter, how much kelp is lost? And what does this disturbance mean for the vast number of species that inhabit kelp forests? These kind of questions ended up motivating my dissertation work, and they're still sort of behind my general interest, my general research direction at UCLA. And in general, you know, in a very general sense, I'm interested in understanding what controls the structure and dynamics of coastal foundation species. So what causes changes in the abundance of important habitat formers like giant kelp? How will climate change impact these ecosystems? What will they look like in 10, 50, 100 years? So I don't know if you noticed, the title of my talk is Macroecology of Giant Kelp Forests. And macroecology is a subfield of ecology that historically has been, you know, really interested in understanding large-scale patterns. This large-scale perspective, it's important. Things like climate change are having large-scale processes like climate change are impacting coastal ecosystems. But I don't want to imply that I'm not interested in smaller-scale processes. And it's become very clear in recent years that small-scale processes definitely have the potential to structure large-scale patterns in ecosystems. And I believe that the field of macroecology has kind of evolved from this strictly large-scale perspective to focusing on understanding the links between pattern and process on a range of spatial scales, and that's really where my interests lie. You know, this multi-scale approach is especially important for coastal ecosystems because they're just so dynamic in space and time. This is a video I made using satellite imagery of kelp canopy kelp canopy cover from 1984 to 2011. And you know, the variability is incredible. Compare this to what you might see in a terrestrial forest over the same time period. It's hard to observe this variability. It requires repeated measurements. And as I'm sure everyone here knows, surveys, especially subtitle surveys, take a lot of time, effort, money. But it's important that we understand these changes. Coastal ecosystems are changing rapidly due to processes like climate change. If you read papers that try to quantify the velocity of climate change, often they find that coastal ecosystems are experiencing the most rapid change. And at the same time, we're becoming increasingly dependent on the ecosystem services provided by coastal habitats. Giant kelp in particular provides a food and habitat structure to a diverse array of algae, fish, invertebrates, marine mammals. I know I don't have to sell this crowd on the importance of giant kelp. In any case, I can never do it as eloquently as Darwin did back in the mid-1800s, so we'll just let his words speak for the importance of these wonderful ecosystems. All right, so let's move on to the specific topics I'm going to address today. And I'm going to use this little story, this short story, to sort of walk us through my talk. So let's start with this first point. You know, there's obviously been a huge amount of research on the drivers of kelp forest dynamics. And I'm not going to even attempt to try to summarize this vast literature. Although I do want to just pick a couple of some of the most important drivers that have been identified by some of this research, right? So first, a number of studies. A lot of this work, a lot of these papers have come out of Paul Dayton's lab down in UC San Diego, have found that large-scale, low-frequency changes in nutrients are the most important driver of giant kelp dynamics. So these fluctuations that occur with climate cycles like El Nino, Southern Oscillation, and the PDO, these results, point to the importance of bottom-up processes in controlling kelp abundance. Top-down effects of predators and grazers are also clearly important, and in some areas, kelp abundance appears to be controlled by urchin grazing, largely. And these observations have inspired a lot of the classic work by James Estes and others on trophic cascades. I have this really basic conceptual diagram of the famous kelp urchin otter trophic cascade, where urchin predators are removed. That causes urchin abundances to increase dramatically, increasing grazing rates, which leads to urchin barrens. But please go see James Estes' papers and books for more informative versions of these conceptual diagrams that talk about interesting things like how the grazing behavior of urchins are influenced by predators and how grazing influences competition among algal species. And then others have sort of argued that wave disturbance is the big driver of kelp forest dynamics. This recent paper by Dan Reed that I actually was a part of, we argued that we compared annual net primary production in Southern California and Central California and argued that the patterns, the differences in annual net primary production were really driven by wave disturbance, more so than nutrients or grazing. The late 90s, Mike Graham characterized the influence of wave exposure on kelp forest dynamics around here, south of Monterey Bay. So we have a good understanding of these drivers, but what we're missing is a really deep understanding of how the roles of these drivers vary in space and time. And I think that this limited understanding is maybe behind some of the past really ardent debates over the relative importance of resource availability, consumer pressure, disturbance in structuring kelp forests. So, I would argue that there's not one right answer. In some places and times, wave disturbance might be most important. Other times and places, resource availability. But again, our understanding of how the roles of these forcings is somewhat limited. And to his credit, Matt Edwards recognized this knowledge gap a number of years ago and conducted these incredibly extensive surveys of kelp forests to try to understand how the impacts of El Niños on these systems vary in space and time. And reading these papers as a grad student really sort of opened my eyes to the need to study these ecosystems across different space and time scales. Just looking at these papers, it just must have been an incredible amount of work. He conducted repeated surveys of kelp forests from Monterey down to Baja California Sur. It sounds like a lot of fun, but a huge amount of work nonetheless. Right? So I have been building on this work, and what I'm going to discuss today is some techniques I've developed to examine kelp dynamics across large space and time scales. And some of the things I've done is compared changes in kelp abundance to spatially explicit data on some of those important drivers of dynamics. So, to put it more clearly, the goal here is to identify where and when a process like wave disturbance is the primary driver of kelp dynamics. Where and when is nutrient availability the primary driver? Where and when is it urchin grazing? And maybe what are some of the other potentially overlooked factors that might play important roles in certain places and times? And a tool I've used to expand these observations is aerial and satellite imagery, so remote sensing data. Giant kelp forms this dense floating surface canopy that's very distinctive when viewed from above, and so it's very well suited to remote observations. And there's been a few studies that have taken advantage of remote sensing data to try to characterize kelp dynamics. For example, a study that's really inspired me came out of Moss Landing. Mike Donnellan, for his master's thesis here, used a time series of aerial imagery in the late 80s and early 90s of the area around Monterey, a little bit south of Monterey, to, number one, characterize this really clear seasonal cycle in kelp abundance in this region. And then two, he did also document some spatial variability in kelp dynamics. He used clustering to identify sort of regions, cluster zones that had similar dynamics. So this motivated my work with satellites, and I started by just sort of reproducing these analyses on larger space and longer time scales. This was difficult until fairly recently because in the past, satellite imagery, the availability of satellite imagery has been low and the cost has been really high. This has really limited the usefulness of this kind of data. But everything changed in 2011 when the USGS and NASA made the Landsat satellite imagery archive freely available to the public. So Landsat is a series of satellites that cover the globe. They've been collecting imagery for over 30 years now. There are millions of Landsat satellite images out there. And so my group has used this data to really create a kelp forest monitoring data set with pretty incredible spatial and temporal resolution and extent. So what we've done is we've taken the 15 tiles, or scenes, 15 tiles that cover the entire coastline from Monterey down to Punta San Hipólito in Baja California Sur, and we've processed the entire archive, all the available imagery for each one of these scenes. So that's over 5,000 images now. There's hundreds of image dates for each one of these tiles. Landsat takes an image of every location on the Earth every 16 days. Again, it's an incredible data set. Because of clouds and other data availability issues, we have an image about every 2 to 3 months of this whole area. But that allows us to look at seasonal, annual, interannual, and decadal variability in kelp canopy on scales ranging from 30 meters, which is the resolution of Landsat, to thousands of kilometers. Now, what we get from Landsat is essentially images. These are basically pictures, and so we need to transform them into usable data, right? So to do this, I've used an imagery analysis technique called multiple end-member spectral unmixing. And what we do here is that, giant kelp has a very distinctive spectral signature, a reflectance signature as compared to seawater, especially in the near-infrared. So giant kelp, like most photosynthesizing vegetation, reflects a lot of near-infrared radiance. And so what we do is model each pixel as a linear combination of pure kelp and pure seawater. So you can imagine these pixels are 30 meters by 30 meters, right? Most pixels are not totally, that we're interested in are not 100% kelp or water. It's a mix of kelp, canopy, and water. And this process allows us to estimate the fraction of each one of those pixels covered by kelp. So we take images and turn them into maps of kelp fraction. And this is obviously just a brief overview. I'm happy to discuss this in more detail with anyone interested after the talk. So we have these maps of kelp fraction. I started this work in the Santa Barbara Channel, one, because I was in grad school at UCSB, but also largely because the Santa Barbara Coastal Long-Term Ecological Research Project there has this great data from these long-term kelp forest monitoring data sets, or sites rather, that was really important for calibrating and validating these satellite maps, for turning these kelp fraction estimates into something more physically and biologically meaningful, and namely canopy biomass. So the LTER divers go out at certain sites every month and measure kelp canopy biomass. What we did is we matched each satellite image up with the scuba diver survey closest in time to that image, and then compared the diver measured biomass at the LTR transects with the kelp fraction of the pixel that sort of overlaid that diver measurement of biomass. So we could compare kelp fraction by Landsat to diver-measured biomass. And we found a relatively strong relationship between kelp fraction and that diver-measured canopy biomass, which we then could use to transform those maps of Landsat kelp fraction to maps of canopy biomass. We looked for an effect of tides and currents on this relationship. We didn't find one. It's possible that there is no detectable effect in Santa Barbara, but other areas that have stronger currents or stronger tides might be impacted. And so I'm currently trying to identify if that effect might be present in other areas. But so far, we haven't seen that. So we have Landsat images. We've transformed them into maps of kelp canopy biomass. And this has provided us with some of the first really detailed regional estimates of kelp canopy biomass on large scales. And we're able to make these estimates repeatedly through time, again, every few months, every quarter, or every season from 1984 actually to the present now. And so now I'm going to talk about how we combined that kelp canopy time series, biomass time series, with some important environmental drivers to try to start getting at what controls kelp dynamics. So this Landsat data has really given us a pretty detailed look at the variability of giant kelp biomass through time. This is a time series of giant kelp biomass summed across the entire Santa Barbara Channel, across this entire Landsat scene, from 1984 to 2015. What this shows is, over this time period, there was a regional mean of about 42,000 metric tons of kelp canopy, but an enormous amount of variability about this mean. Generally, you can see a seasonal cycle with lows in the winter and highs in the summer or fall, but there's a huge amount of variability in the magnitude and timing of this seasonal cycle. Then that annual cycle seems to be superimposed on a sort of longer period, 11 to 13-year cycle. In this region, we didn't see any long-term secular trends in kelp canopy biomass. So we compared this data to sea surface temperature data, which we're using as a proxy for nutrient concentrations, and wave heights collected from a buoy off of Point Conception. So this is just a single, this forcings data is just coming from a single point. So we're just looking at how sea surface temperature and waves are associated with regional changes in canopy biomass. So we found that both waves and nutrients help explain that seasonal cycle, right? So the figure on the left shows this. We looked at percent loss over the course of each winter, percent canopy loss over the course of each winter, compared that to the maximum wave height during that same winter, right? And this is showing this saturating relationship between wave height and winter canopy loss. Now, importantly, this only holds for extreme events. So if you look at mean wave height over the winter, this starts to fall apart. So extreme events are important here. And the figure on the right shows how lower sea surface temperature, which corresponds to higher levels of nutrients, is associated with higher growth rates. So lower sea surface temperature, higher growth rates. So it appears that at least in the Santa Barbara Channel, waves are associated with loss and nutrients appear to influence recovery. Now, when we start to examine dynamics on larger scales, it becomes really clear that there's a lot of variability spatial variability in kelp dynamics. This top graph shows the regional canopy biomass for Central California. You see this very clear seasonal cycle, not much interannual variability in kelp canopy biomass. Down in Southern California, again, there's a seasonal cycle, but it's a lot more variable. You also see this long-term, again, that long-term 11 to 13-year cycle. So these next figures show, I think, that seasonal cycle a little bit more, that spatial variability in seasonal cycle a little bit more clearly. So this gives the mean and standard error of kelp canopy biomass for a couple locations around Southern California. The mean and standard error for each month over the time series from 1984 to 2015 for these sites around Santa Barbara. And what you see is in the area off Point Conception, San Nicolas Island, you see that very clear seasonal cycle. Maximums almost always occur in the late summer, early fall. minimums in the winter or early spring. And there's not much variability in that timing from year to year. That's shown by these small error bars. In contrast, the Santa Barbara and Malibu show a lot of variability in that seasonal cycle. So these large error bars indicate that those maximums and minimums aren't occurring at the same time each year. And the variability in this seasonal cycle, at least in Southern California, appears to correspond to wave exposure. The yellows, in this area, we get our big swells in the winter, these west-northwest swells. The yellows and greens represent high wave heights for just a example, a typical west-northwest swell. And so those exposed areas, they are the ones that have this clear seasonal cycle. Kelp gets wiped out when these swells start hitting in the fall and early winter each year. It recovers pretty reliably in the spring. The cycle is a lot more variable for these sheltered areas, the dark blues here. It takes a really big swell to have a large disturbance in these regions, and so they don't lose their canopy each year. Okay, we haven't yet really got to quantifying how the most important drivers vary in space. And so to get a clearer picture of what causes spatial variability in giant kelp dynamics, we used a general additive models to identify the relationship between kelp canopy and predictor variables for all of Central and Southern California. And I want to mention that this is coming from some work led by Tom Bell, some paper he has currently in review. The general concept of this generalized additive model is it lets you model a response variable. In this case, we're talking about giant kelp canopy biomass as the sum of nonlinear functions of different predictor variables. And so these are predictor variables. I'll mention them in a bit. What we've done here is we've broken up the coastline into 500-meter segments and modeled each segment individually, separately. Right? There's 732 of these 500-meter segments that have a long enough Landsat time series to run this analysis. I want to describe the bottom panels. This is showing mean canopy biomass and the coefficient of variation of canopy biomass along the longshore of the study area. I'm showing this because a couple of the later panels are in the same format. So this goes, the y-axis here shows that our sites are segments, coastline segments from south to north, starting in San Diego up through Santa Barbara. These dotted lines are the Channel Islands. And for each Channel Island, we start at this pink arrow and then go around clockwise. So at the end of these dotted lines, we're picked back up at Point Conception, go up through Monterey. So on to those predictor variables. We were able to get spatially explicit data on many of these variables we hypothesized had an important influence on kelp dynamics. Maximum wave height came from this model run by the Coastal Data Information Program. We were able to extract daily wave data for each coastal segment. All of these data, so the giant kelp biomass data was placed on a quarterly time scale, and so we put all these predictor variables on the same quarterly time scale. For wave height, we're looking at the maximum wave height over a quarter. Again, we have a different time series for each coastal segment. To get at mean surface nitrate, we're using satellite imagery-based sea surface temperature and a relationship relating sea surface temperature to nitrate concentrations. So again, spatially explicit, we have a separate time series for each coastal segment. The NPGO index, this is not spatially explicit, so each site gets the same time series of NPGO. This is positive values in this index correspond to stronger wind-driven upwelling, so it leads to greater nutrient concentrations in some parts of the California coast. We included this kelp occupancy term. This is just biomass from the previous quarter or season to account for temporal autocorrelation in the time series. Then ISP Alginets was a kelp harvesting company that harvested canopy in certain sites up until around 2006. And so we used their records to add this harvest effort term. Then finally, for a small fraction of the sites, I believe, let's see, right, 45 of the 732 sites, we did have a time series of sea urchin density. So for those sites, we included sea urchin density in this analysis. So we lagged each of these predictor variables by 1 season. So we're using predictor variables from the season prior to predict canopy biomass in the current season. There's a lot going on here, I know, so we're going to step through each panel one at a time. Again, we ran separate models for each coastline segment, so we can identify how the primary drivers of kelp dynamics vary in space. Again, y-axis for all these panels shows the coastline sites from south to north with the Channel Islands as dotted lines here. The upper left panel, this shows the amount of variance explained by the GAM, by the generalized additive model, for each coastal segment. So on average, these models explained about 0.4, the fraction of variability in kelp biomass explained was about 0.4. But you can see that in some locations, the models explained a lot more variability. Other locations, the models didn't do as great a job. And this is an important point I'm going to come back to in a slide or two. So the rest of these panels, B through G, they show how the relative coefficients of these potential predictor variables vary in space. So these model coefficients show the strength of the effect and the direction, right? So let's start with maximum wave height. So what this is showing is that maximum wave height had a strong negative effect on kelp canopy biomass north of Point Conception. So in most of these sites, maximum wave height was the most important predictor, was the primary predictor of giant kelp biomass. As you move down to Southern California, that effect lessened a little bit. Waves had a lesser impact, a lower impact on kelp canopy biomass in Southern California, except for some of these exposed sites. Nitrate concentrations, as estimated by sea surface temperature, they tended to have a positive effect on kelp biomass throughout the study area. They only were the primary, the largest contributor toward the model for certain sheltered sites in Central and Southern California. Then the positive deviations in the NPGO had a positive effect on giant kelp biomass for some sites in Central California, but generally NPGO was not the primary driver, the most important predictor for the sites. Kelp occupancy had a positive effect on kelp biomass, pretty much for the whole study area, and this makes sense. If kelp biomass is high at a given point, it's more likely to be higher in the next quarter. Again, this helped account for some of the temporal autocorrelation in these models. Harvest effect had pretty small effects. Harvest effort had a pretty small effect on kelp canopy biomass. It typically was not a primary driver. Then sea urchin density. Again, we only had sea urchin data for a handful of sites. For some of the sites, for about 25% of the sites that we did have it for in Southern California, it was the primary driver of kelp dynamics. And however, in Central California, it was really not related. It wasn't related to kelp canopy biomass for most locations. So these plots then show the nonlinear effects of each predictor at the sites for which each was significant. And so this gives you an idea of sort of the threshold. This tells you, so the areas above the 0 lines are areas where the predictor had a positive impact on giant kelp. The magnitude of the effect is characterized here on the y-axis. Note that the y-axis, the scale of the y-axis is different for each panel. So for example, wave disturbance shows this really large negative effect on kelp canopy biomass, once you get above about 2.5 meters wave height. So this gives you some idea of some of the thresholds of these variables on kelp biomass. Once you cross that threshold, there's an increasing negative impact on kelp canopy biomass, which makes sense. Nitrate, once you get below about 2 micromoles per liter, there's a large negative effect. Again, this is useful for identifying some thresholds in the effect of these predictors. And then nitrate had a positive impact on kelp biomass, or was positively correlated with kelp biomass at levels higher than about 6 micromoles per liter. Let's skip down to sea urchin densities. This was always negative impact. It seems to saturate at higher sea urchin densities. So earlier I mentioned that these models didn't always do a good job of capturing a lot of the variability in kelp biomass dynamics. And the details on where and when these models don't perform well is really useful for identifying other potential drivers of kelp dynamics. So for example, this dotted line shows the modeled kelp canopy biomass for this site that contains Carpinteria reef here. This is the reconstructed from the GAM, the modeled kelp canopy biomass. This model, urchin densities were not included in this particular model because the time series wasn't long enough. But we did have some urchin data starting around the early 2000s that we could use to examine why this didn't fit very well in a certain time period. So the dotted line, that's the modeled kelp biomass. The solid line is the observed Landsat kelp biomass. And up until 1998 or so, this does a reasonable job the model does a reasonable job of predicting Landsat, predicting kelp canopy biomass. But after that 97-98 El Nino event, the model predicts a relatively quick recovery of kelp biomass and then continued fluctuations. But kelp did not recover here until about 2005. And so this area of model misfit is explained because during this period there was this very high densities of sea urchin, which caused a large persistent urchin barren at this site. So this spatial analysis of model misfit, I think, is a potentially useful tool for identifying other underappreciated or unknown drivers. I think there's a lot of promise for examining the spatial and temporal patterns of this mismatch across the California coast. All right, so to summarize those environmental drivers, the seasonal and longer-term cycles of giant kelp biomass are really variable, highly variable in space. Waves seem to be dominant driver in exposed locations. Nutrient and urchins are the primary driver in more sheltered locations, especially in Southern California. So I'm going to finish by I've been discussing environmental drivers. I want to briefly talk a little bit about spatial drivers. So there's a large body of work on how strictly spatial factors influence population dynamics of metapopulations. Now, metapopulation is a group of spatially separated subpopulations that interact at some level. Usually this interaction occurs through dispersal of individuals, larvae, spores. And there are some requirements that a system has to meet to really be considered a metapopulation. It's useful to see if kelp fulfills these requirements. One, you have to have patchily distributed populations. Otherwise, you're just having a normal single population, and this framework isn't very useful. Giant kelp forests are indeed patchy. A major giant kelp generally requires hard substrate between 0 and 30 meters, and since hard rocky substrate in California tends to be patchily distributed, so do kelp forests. Secondly, you need to have interaction between these subpopulations. Otherwise, again, they'd just be independent populations. There would be no need to understand the spatial arrangement of these patches. Generally, they interact through dispersal, again, individuals, larvae, et cetera. And giant kelp is not mobile as adults, but subpopulations of kelp do interact through the dispersal of spore stages. And some empirical and modeling studies have indicated that this dispersal occurs on the order of meters to kilometers, which is sufficient to far enough that spore exchange occurs between patches. Finally, subpopulations need to have turnover. and somewhat independent population dynamics, right? If all of these subpopulations had the exact same dynamics, again, you just treat them as a single population, there wouldn't be the need to understand the spatial arrangement of patches. And there's a lot of similarity in the dynamics of patches of giant kelp, but if you look, that are close together in the same area, but if you watch this video, you will see that certain patches go extinct temporarily while others persist. These dynamics are not completely synchronous throughout this region. Okay, so it seems that giant kelp is a metapopulation. It seems like it fits these characteristics. So why would we care? Well, I think one major reason we care is because there's a rich amount of metapopulation theory that we can use to try to provide insight on how the arrangement of patches of giant kelp might influence its dynamics, its persistence, the frequency of recovery from these disturbance events, right? So, you know, if you look at this area or think back to this movie, all these patches are generally experiencing the same wave exposure, more or less. They're experiencing similar nutrient conditions, but we do see variability at smaller scales. And so some of this variability might be, again, due to some of these demographic connectivity. So metapopulation theory would predict these two hypotheses. One, larger patches should be less likely to go locally extinct, and they should recover quicker from these temporary extinctions. Secondly, more connected patches should also recover from extinctions. So we were able to test this with this great Landsat data set. We have basically complete coverage of this area. We can measure temporary extinctions and recoveries and test some of this theory. So here we used the time series for each patch to document extinctions and colonizations, right? So the colors represent distinct patches here. They don't have any other meaning or scale. Part of this work is developing a generalized way to define patches and metapopulations using network theory. There's not time to discuss this now, but happy to I'm really a nerd about some of this network theory, so I'm happy to talk about this later for anyone interested. But anyway, these are the patches we defined. We tracked them through time. If any part of a patch had kelp canopy in it, it was considered occupied. If the patch was totally, remember, think of these as habitat patches. They're built from composite kelp canopy. So we're thinking of this as total available habitat. If when biomass for the entire patch fell to 0, we considered it extinct, temporarily extinct. We set a rule that these patches had to be extinct for at least 6 months, had to be 0 for at least 6 months to be considered extinct. That way, so if there was subsurface canopy that survived a wave event, say, we would expect that it would grow back within 6 months. So little blips where it drops down to 0 for just a quarter or just a few months, we weren't considering extinctions. Then, when a colonization occurred, when a previously extinct patch showed kelp canopy again. So with this data, we have identified all the local extinctions and colonizations for each one of these patches on that entire Landsat data set. So we were able to calculate the fraction of time each patch was occupied. We were able to calculate the average probability of extinction when there was kelp at that patch, the probability of recolonization when kelp was absent from that patch. Then we compared occupancies and this extinction and recolonization probabilities to patch size and connectivity here. So patch size is pretty easy to understand. Again, that's just the size of available habitat, the size of suitable substrate. That does not change through time. Patch connectivity, demographic connectivity, was a little bit more complicated to calculate. Some folks, there are indices that just use Euclidean distance, and that's fine. But we went a step beyond and tried to calculate connectivity, demographic connectivity, based on source fecundity and ocean transport. So the goal here is to make an estimate of the probability of kelp spores moving from patch I to patch J. For a focal patch, let's say patch J, then we sum all the connectivities of its neighboring patches. So I'll try to explain this. So there's three parts of this: the source fecundity, ocean distance, the transit time, and then spore loss. So for source fecundity, we first found that estimates of kelp canopy biomass are a relatively good predictor of local population fecundity. That's the number of spores or sorus area per meter squared of reef. So that's this biomass term of patch I. So again, patch J is our focal patch. Let's say that's that cyan patch here. Patch I is a neighboring patch. So the biomass of patch I gives us an idea of its fecundity. Then we used Lagrangian particle simulations to try to understand the average amount of time it takes spores to go from patch I to patch J. This is based on these ROMS models. So this is transport time. We'll call it ocean distance. And then finally, we accounted for spore loss during this transit time using spore loss functions. We tried a number of different spore loss functions. We did a sensitivity analysis to make sure that our results weren't too sensitive to that choice of spore loss function. So anyway, again, this gives us essentially the probability of a kelp spore arriving at this focal patch from a neighboring patch. To get the total connectivity of this patch, we sum all of those J to I connectivities. This figure just shows the index of connectivity. So these tightly grouped patches, as you might expect, have higher levels of connectivity. So to summarize this, we then use logistic regressions to try to look at the relationships between pack occupancy, probability of colonization, and probability of extinction, compared that to patch size and connectivity. For each of these plots, patch size is shown by the colors. So we group patch sizes into three different categories. These blue dotted lines are the smallest patches, the green dashed lines are the medium-sized patches, and the red solid line is the largest patch. Then x-axis give relative connectivity. So starting on the left side plot, we find that these largest patches have a very high probability of occupancy. They're usually occupied. And so connectivity doesn't make a big difference for them. Whereas for the smallest patches, connectivity makes a big difference in terms of probability of occupancy. So as connectivity increases, probability of occupancy also increases. This middle plot shows probability of colonization. So when a patch is extinct, how likely is that patch to recover? And for all three patch sizes, increase in connectivity increased the probability of colonization. Now, at a given connectivity level, the larger patches were more likely to recover. Then the last plot on the right, this shows the probability of extinction. If a patch is occupied, how likely is that patch to go extinct? And this kind of mirrors the probability of occupancy. Those large patches have a very low probability of extinction, whereas the smaller patches, relatively high probability of extinction for small isolated patches. As connectivity increases, the probability of extinction decreases. All right, so to summarize this, let's start with the relationship between that probability of colonization and patch size and connectivity. So our interpretation is that larger patches are more likely to colonize because they represent a larger target for colonizing spores. The well-connected extinct patches are more likely to recolonize because they're more likely to receive colonizing spores from neighboring patches. There's more spores in the area for a well-connected patch. So, but why would a larger patch be less likely to go extinct? Well, we suspect that larger patches can, on average, support larger populations. So these larger populations just simply have a smaller chance of going fully extinct from a disturbance event like waves. It's also possible that these larger patches have more variability in small-scale features like bathymetry, slope, and that these can serve as maybe refugia from certain disturbance events. Now, it's a little bit harder to explain why more connected patches would be less likely to go extinct. One hypothesis we're testing is that inbreeding depression higher levels of inbreeding in small, isolated, old patches reduce fitness of kelp via reduced growth and reproductive capacity. And so this might cause these isolated small patches to be more likely to go extinct. So to summarize these spatial drivers, we've linked our comprehensive satellite imagery-based data set on giant kelp population dynamics to some predictions of metapopulation theory. And we've found that those large, well-connected patches are more occupied more often. They're less likely to go extinct and more likely to become recolonized. So, to summarize, as is usual, these results just sort of open up more questions. And there is a lot of work to do to sort of integrate these spatial and environmental drivers. Just for one example, I'm interested in looking down at close to the range limit of giant kelp. And some folks have observed that kelp populations down in Baja take a lot longer to recover from disturbance events like El Nino. So one question is how much of this delayed recovery is due to warm sea surface temperatures and potentially nutrient limitation down there? How much of it is due to spatial factors? Those kelp forests down in Baja tend to be more isolated. There's often large amounts of space in between patches down in Baja. So how much of it is environmental? How much of it is due to this limited population connectivity? So I'm just about done. I've talked a lot about trying to use new tools to better understand kelp dynamics on large scales, but I've only talked about California, right? And so I'm going to end today by just mentioning that program that Mike introduced, the Zooniverse Program. The goal of this is to try to do what we've done in California on global scales. Now, that California project took a number of years of effort, and so it would take a very long time to process that Landsat satellite imagery for the rest of the world, for Chile, the southern islands, Tasmania, New Zealand. And so we're launched this citizen science effort, Floating Forests, to try to crowdsource identification of satellite imagery. And it's been a success so far. We've had almost 500,000 of those Landsat images, those Landsat tiles, completed already. This is in the face of some real hurdles we've run into due to image quality. But it's moving today. This is really interesting. It's really fun. The interface is simple. You don't have to know what you're doing to help, to have a good time. You basically just use your mouse to outline kelp forests. It's a great outreach and learning tool. We've already noticed some pretty dramatic declines in kelp abundance around Tasmania. And so we hope to use this data to understand some other interesting patterns in less understood regions. So if this sounds like an interesting project, please spread the word. If you're interested in getting involved, we'd love to have any more help on this. You can use this in the classroom. We're creating a number of lesson plans that sort of take advantage of this project. Check out ZooTeach for lesson plans based on floating forests, but also based on some of the other Zooniverse projects. They have projects using to look for galaxies out in outer space. All this data will be freely available very soon, so you can play around with kelp dynamics. I should also say all of that Landsat data that I discussed today is now freely available through the Santa Barbara LTER. So if you're interested in using it, you can get it from the LTER. I'm happy to help if you think it might be useful for your project. It's a little bit of a headache to deal with, but once you learn some coding techniques, you can figure it out. So yeah, please spread the word and contact me if you're interested in getting involved in this or have any questions about this project. So that's all I have. Thank you very much again for invitation and for your attention. Appreciate it.
Questions?
Sure.
So you're using Landsat. Is that a multispectral platform?
It is multispectral.
Are you getting? Can you distinguish? You said you're using reflectance to
Right.
get your kelp data. Can you distinguish macrocystis from nereocystis?
We can't distinguish macrocystis from nereocystis. So Landsat is a series of satellites. The longest times, the real long time series is coming from Landsat 5, which was launched back in 1984. With Landsat 5, this is really made for terrestrial remote sensing. We're working on right at the edge of what's possible with detection limits. So with Landsat 5, we can get macrocystis. It seems like we're not capturing nereocystis very well. We're only getting at the most dense times. Landsat 8, same spatial resolution. This was launched in 2012. It has a lot higher signal to noise ratio. So with Landsat 8, it does seem like we can pick up nereo. So we can't differentiate nereo from macro, but we can at least pick up nereo. We're interested in trying to, at least for areas where we know this is nereo or macro, we can start doing time. But you know, with the multispectral, the core spectral bands, we can't differentiate that. NASA is planning to launch a hyperspectral satellite with similar resolution to Landsat. They say 2020, but 2025. So in 10 years, we should be able to start doing some of that.
How do you know for a fact that a giant kelp population is extinct? Because there's been plenty of times where I've gone diving and you don't see any kelp canopy,
Yeah.
but there is kelp there.
Right. So the short answer is that we can't be sure that it's absolutely extinct. We can't pick up. So with this method, we're only able to see surface canopy. So if there is subsurface canopy, we're not going to capture that. If by using that sort of 6 month or expanding that to maybe a year definition for an extinction, we can at least account for periods where there's some subsurface canopy that reaches the surface. But you're right, for detailed genetic analyses, we cannot, there is the detection limit is an issue. We're not picking up subsurface canopy. We also might be missing small, sparse kelp beds. So there is some sort of detection floor with Landsat. Again, that detection floor is lower with the new Landsat 8, but it's possible that we're missing some of the small, sparse beds. So keep that in mind when you're trying to link this to some genetic analyses or trying to really get into details about demographic connectivity. Absolutely, it's a good point.
So you guys have looked at some of the metapopulation dynamics, off San Diego and now up off Santa Barbara, maybe some other regions. I'm just curious if the patch dynamics seem to be different regionally where you've done this and what the different drivers might be.
Yeah, that's a great question. We're just starting to look at that now. So most of that metapopulation work, we didn't look at variability within regions, but Max Castrani, the postdoc at UCSB, he's doing that right now. Actually, might have, I don't have a slide, but he's asking that question right now. It's an interesting one.
If I understand your graphs correctly, you were comparing nutrients to biomass, but you were assuming nutrients based upon winds. Have you looked at the actual relationship between patchiness of nutrients and winds?
So we were using sea surface temperature rather than winds, and so we've used those published relationships between sea surface temperature and nutrients. Have not incorporated winds into that yet, but there's certainly a lot of room for improvement to that model, especially as we go to these really large spatial scales where we're not totally sure that relationship will be the same. So that's an area we're working on, but we have not incorporated the winds to that nutrient calculations yet.
So to follow up on that, the relationship between temperature and biomass that you showed looked like it was driven by two data points that had really high temperatures and lower mass. So is there very much of an effect of temperature itself without the nutrients being thrown into that?
So that's a good point, and if we're looking back to the GAM model, we sort of see the same thing. I think that it was driven by those two data points because it's a threshold response. So it's sort of like most of the time it's not limited. Once it goes into limitation, that probably reduced growth rates a great deal.
But it's temperature itself.
Right, temperature itself. We have not been able to tease those two things apart. Again, because we're using sea surface temperature to infer nutrients, I have not been able to sort of extract those two different dynamics. But that's something that I'm really interested in looking at down in Baja too, where we get warmer temperatures. And so, you know, I think we're going to need nutrient data to get at that.
So a lot of what you've looked at has kind of confirmed, you know, in specific regions that certain things are important that we kind of already knew were important.
Right.
We knew waves are important in nutrients, et cetera. And then when you actually get to the metapopulation model, one of the bigger things that comes out is that patch size really is important to both the recovery and the ability to not go extinct. You're now working at a scale where you could potentially help inform us as to what drives patch size. We haven't been able to do that before because we generally work within individual beds. So if patch size is so important, do you have any ideas about what actually drives why some beds can become larger, much larger than others? Or does this become circular because the more wave exposed you are, the smaller you will be and you'll never, I mean, can you do that with this technique or?
You know, I wonder a big part of patch size might be sort of substrate and bathymetry. And so I think if we could get, so far we've used, again, composite kelp canopy to sort of infer substrate. So one thing I'd like to do is if we could get better substrate data, bathymetry data, then we could start looking at areas where, you know, compare areas where kelp canopy had filled up the available substrate to areas where it had not filled up the available substrate. And I think that goes a long way towards answering that question. So with what we have right now, we haven't been able to look at that, but with some, and there is better substrate data that we can start using to get at just that. It also brings up another important question about how you define a patch, and there's a whole another story about you know, what do we call a patch? Because this huge area of rocky substrate might really be supporting multiple interacting populations, especially if that whole area doesn't get occupied because it's disturbed often or what have you. But that's an interesting point that we're.
All right, well, I'm going to let Kyle off the hook right now. We got time.